Let's start up here. Let's all focus on immunology and let's gather our thoughts. Why is everybody so agitated? Yeah. Hi.
Or yes,
I think I posted all of them. I will check it out after class,
okay.
just in case. But I'm pretty sure I did. I hope in the right class. Okay. All right. So everybody's ready? It's a tough class today. I know it's a lot of details, and I just uploaded the, updated PDF with a little bit more details. Yay! No darts towards this side. Good. Okay. Yeah.
I see what you said.
Details that is that good? So details that are important for you to understand a little bit more on what happens here with this lymphocyte development. Okay. So let's just get into it. This slide we had before, but I realized we had a bit more wording than I expected to have to make sense. So I just put it here again because this is very important. Okay, and remember, if I say something is really important, so for sure I'm going to ask you about it in the test, right? So everybody's awake? Yeah, yeah. So what is important here is this transcription factor that is called AIRE, okay, AIRE, and this is happening at the thymus. Remember, we're wrapping up with the thymus, with the T cell development, right? So this is happening in the thymus. And the question here is, what are the cells that are expressing AIRE? So you have two cell types in the thymus that express this transcription factor. One of them is in the medulla of the thymus, so you call them medullary cells, but they're in the thymus, so you call them medullary thymic cells. And they happen to be epithelial cells. So that's a long name for those cells. Medullary thymic epithelial cells. And the acronym here is MTEC. Okay, but you think that they're medulla in the thymus, and they are, if you find some medullary thymic cells, those are most likely the ones that are expressing AIRE. Besides those cells, you also have these other cells that are dendritic cells in the thymus. Okay, they live in the thymus. So those dendritic cells that are represented here, they're also expressing that AIRE. And what is the AIRE doing? Transcription factor that is going to a lot of genes, basically all of the genes that you have in your body, they're expressing proteins. Okay, genes that are expressing proteins as the gene product. So this AIRE will be expressing little short pieces of proteins from all of the genes in your cell. Okay, so all of the genes in your genome that are expressing some kind of proteins, and those genes are basically representing proteins from all over your body. Okay, so all over the body you're going to have little pieces of proteins, either 8 amino acids long or 12 amino acids long. They're going to be expressed because of this AIRE transcription factor. And that is going to make all of these antigen or self-antigens buffet for your single positive thymocytes. Right, so this is the stage that is not double positive anymore. So this guy is a single positive. Okay, so what's going to happen? If your thymocyte recognizes something on this self-antigen buffet, then the thymocyte is going to be killed, right? So that's your negative selection. Okay, make sure you understand there is a difference between negative selection and positive selection. Okay. So the thymocytes that do not recognize a self, they are then allowed to go out to the periphery. But remember, those thymocytes already had been positively selected. Okay, so not only they are not going to recognize self, they are also not going to interact with B cells unless they have to. Okay, so they're not going to interact with those MHC molecules unless they have to. Okay, so let me see if this is all okay. These are all the important parts here. Do you have any questions about that? Cool with that? All right. So here I have the example of a syndrome that happens in case you do not express the ARI gene very well. So if you have mutations in that transcription factor, now you are not going to be able to express some of those proteins that are important for that self-antigen buffet. That means you're not going to teach your T cells what is self and what is not self. So the T cells that are coming out of that thymic school, some of them are going to be self-reactive, because when they go out in the periphery and they start looking for whatever they can bind to, they can bind to things that they did not see in the thymus, right? So if that transcription factor here in the thymus is not working properly, you're gonna start having a self-binding T cells, and you're gonna have a lot of problems, okay? Because this is an autoimmune disease, okay? And this disease, when the ARI gene is not expressed properly, this is called autoimmune polyendocrine. So you know endocrine system is very much involved in this, right? Polyendocrine syndrome type 1, or APS1. There's another name for it. It's autoimmune polyendocrinopathy candidiasis ectodermal dystrophy. Yeah, try to say that 5 times quickly. APECED, like the whole alphabet you put in there. Just remember APS1. Okay, that's a problem with the ARI transcription factor. And here I have little Kayla. At this time she was about 12 years old, and she was going for Miss Broward pageant something. So she has something to say.
When I want to be a pastry chef, I do dolphin therapy every year, which is where I get to swim with dolphins. I have a genetic disease called autoimmune polyglandular syndrome type 1, where my lungs and ovaries and organs and endocrine glands attack themselves. I wish to be in this Broward pageant to advocate for girls with hidden disabilities and silent diseases like me.
Okay, so that was Kayla, and she's a daughter of a friend of mine. And she's doing okay. She's about 16 now. Actually, a few weeks ago, she was in some of those local news channels, also advocating for APS1 children. And she's doing quite well, remarkably well, considering, you know, if you look up what this disease encompasses, it's very scary. Okay, so here, development of your T cell. Important things that you have to know. Yes, this thing looks scary, but it's supposed to make it as simple as possible. So what are the things that you have to know here? You look here at your T cell being developed. First thing that happens, you're going to have still in the stem cell stage, you're going to have a cell that is not fully committed to T cell, but because of the presence of some transcription of factors and the expression of some CD markers on its surface, then this precursor cell is going to be committed to becoming a T cell. One precursor that you should know here is, well, it's not here, but is interleukin 7, which I had mentioned before, right? Interleukin 7, and also c-KIT. Okay, so those two are important for the commitment to your T cell. There are also other factors, but here we just stick to those for now. Okay, so what's happening here? At your lymphoid lineage committed precursor cell, not much is happening. Okay, the only thing that is happening at this stage over here is expression of a CD marker. You have the expression of c-KIT and interleukin 7, okay? And nothing else really is happening except for rapid cell proliferation. Okay, this is in the bone marrow, okay? So the cell did not come out of the bone marrow yet. So the cell is proliferating. It is a precursor cell that is going to go to the lymphoid lineage, but it didn't quite decide yet if it's going to be a T cell or a B cell. Okay, you could just stay in the bone marrow and become a B cell. Okay, so it's kind of undecided. But the first thing it has to do, it has to proliferate. What happens when a cell proliferates? It is dividing. What is the process? What is the first thing that is happening for the cell to divide? Yeah, it is one of the growth stages.
Is it called?
Oh, this guy is undecided. It didn't even get to the stage yet. This is a baby, like it's a stem cell, a precursor cell. It's going to divide just for cell division. Everybody took cell bio? Yeah. How does the cell divide? It's just a simple question. Don't go complicated. Got chocolate. Mitosis, yes. What happens in mitosis? Copy the DNA. Ready? Okay. Good catch. Ready? You don't want to, no chocolate. All right. All right, mitosis. You're going to copy the DNA. You're going to make a copy of the DNA. Semi-conservative replication. Remember all the stuff? Yeah, don't forget about it. It's important. The cell's going to divide, right? You remember the enzymes that are working for replication of the DNA? What do you call them? DNA polymerases, right? So what are they doing? Getting nucleotides and going there and matching nucleotides and purines and pyrimidines and checkpoint and blah, blah, blah. Now, when the DNA is dividing, you don't want to mess up the cell. It's still a progenitor cell. It has all of the genes that all of your other somatic cells have, right? Now, when the cell is dividing, you definitely don't want to start upregulating some genes that can mess up and rearrange things around, because then it would be a mess and your cell would die because it would become transformed, right? You just want to duplicate your DNA best way possible, right? So you have all these proofreading mechanisms, and you just want to make sure that your daughter cells are as similar as your parent cell as possible, right? So look at this. Your rearrangement genes are not being expressed right there when the cell is replicating, okay? Your TDT genes, okay, that's not TNT, that sounds kind of explosive, right? Now, this guy is terminal deoxyribonucleotide transferase gene, is going to add nucleotides to places that didn't have any nucleotides there. It's like, what do you call it, template independent. Okay, that guy is not active. Neither of these two guys are active. Okay. And then the only thing that is happening here is really basic transcription factors that committed this thing to lymphoid lineage, right? And that's pretty much it, and cell replication. That's it. Then what's going to happen? You're going to have expression of a particular cell marker that is going to tell this cell to become a T cell, okay? And here is that marker. CD25, okay? So now the cell becomes CD25 positive. It leaves the bone marrow. Look, it's not in the bone marrow anymore. It leaves the bone marrow and it goes to the thymus, okay? So expression of your CD25 is going to allow this cell to get to the thymus. So we know this cell is going to become a T cell, okay? The other factors too, but let's focus on this one, okay? So now, the cell gets to the thymus, okay? And yeah, it was still like proliferating, okay, still proliferation going on. It gets to the thymus and then it stops proliferating. Look at this, it doesn't have anything on the surface. It's very much naked. Well, it has some of those like CD25, but nothing to do with the T cell receptor on the surface, okay? No T cell receptors there. So it gets to the thymus. Now, in the thymus, it's going to start rearranging the genes, and is going to start adding some nucleotides to some joining sessions. And this is going to happen only at, which one is the chain that starts to rearrange first of the T cell? The chain, which one? You have two chains for your T cell receptor, right? You have a beta chain and an alpha chain. So the one that starts to rearrange first is the beta chain, is the longer chain, right? So this gene here, that is TDT enzyme, it is only going to be expressed when your beta chain starts to be rearranged. That's the only time here for your T cell that you're going to see the expression of that gene. Okay, so your heavier or beta chain is going to have more variety and possible, differentiation than your alpha chain. Because see, when you go here and you see a development of the alpha chain, you don't have that gene being expressed. So this here is the only time that you're going to see that gene expressed. Now, besides this guy, you're also going to have your RAG expression, right, between your pro-T and your pre-T. You're going to have the RAG, right, rearrangement of those genes that you can pick and choose from, right? And you're going to see more mechanisms of how that's happening, okay? So you have both of those guys, RAG and TDT being expressed, okay? And now you're going to have to, you're rearranging your beta chain until you find here, rearranging right here, okay, until you find a very good combination that is going to allow your beta chain to be put on the surface, okay. So look at this. This pro-T cell looks very much like that progenitor naked cell, but here you have rearrangement of the genes happening. Once you get that beta chain on the cell surface, then this thing becomes a pre-T cell. Okay, so it's not pro-T anymore. Now it's a pre-T. It's a pre-T cell because it looks so pretty. No, it's a pre-T cell because it has a beta, but it doesn't have an alpha. Remember, we have to first test the beta chain in order to make sure it works. Then you're going to start recombining the alpha chain. Okay, but then in order to test the beta chain, you need the alpha chain. So that is the chicken and the egg problem, right? And the pre-T cell solves that problem by expressing that pre-T receptor, right? The pre-T, what do you call it? Pre-T protein, pre-T alpha chain, right? Pre-T alpha chain. So this chain here is, okay, right there, pre-T alpha chain, right there, alpha, the pre-alpha, okay? That chain is not variable. That is a gene that is the same for everybody. Everybody expresses exactly that gene because you just want to test whether your beta chain works or not. So at the pre-T stage, you're going to see that. You're going to see beta and pre-T alpha, okay? Now that you tested your beta chain and it works, so you say, okay, now I'm going to make the alpha chain. And before you make the alpha chain, since your beta chain works, you want to make sure that you're going to get a lot of cells that have that specific combination of beta chains. So what do? First thing you do, you turn off your recombination genes. Okay, especially this one gets turned off and it does not get turned on again. Your RAG 1, 2, those get turned off because again, your cell is going to undergo proliferation. The cell went to a lot of trouble to make the beta chain. Now that it's successful, you're going to make a lot of daughter cells that have that beta chain. Then after that, each one of those daughter cells is going to start to differentiate your alpha chain. So you're going to have a lot of different cells that have the same beta chain, but then different alpha chains. Okay? So when you do the proliferation, of course, you don't want to mess up with the DNA replication. So those genes get turned off. Okay? So proliferates. Now you're going to go to your alpha chain recombination. Okay, so this is not further subdivided. But after you proliferate, you stop proliferating, now you're going to recombine your alpha chain. Okay, at this stage, when you get beta and alpha on the surface, you're also going to get CD4 and CD8 that are going to be expressed. Okay, and you're also going to get CD3. That's a marker for T cell. Okay, is a part of your T cell receptor. So you're going to get, this is your double positive stage. You have T cell receptor that is working. You're going to have CD4, CD8, and CD3. Okay, right there on the surface. So at this stage of your double positive stage, all your genes here are going to be shut down because your chains are working. You're not going to be recombining anything else here for your T cell. And this is one of the differences between T cell development and B cell development. Because as we see for the B cell, you can further recombine things down the line here. Okay, but for the T cell, you don't. Okay, so this is stop recombination. Then what's going to happen? You're going to have here your testing of your T cell. You're going to have your positive selection, right, in which you're going to detect either one or the other. You're going to keep it there. Then depending which one, CD4 or CD8, that is going to interact with the MHC, you're going to have your single positive stage right there. Single positive also has CD3, okay, CD3 right there, okay. And at this stage, you are going to see whether you have activation due to the self-antigen buffet. So right after you have your positive selection, you're going to have your negative selection. Okay, anything that reacts to self is going to be killed. If the cell survives, it passes all of these tests, then it's going to come out of the thymus and is going to go to the periphery as a mature but naive T cell. Okay, so make sure you know this is not mature, naive T cell. Okay, naive because it has not seen the real antigen out of the thymus. Okay, it has not seen antigen that is foreign to the body. Okay, and it's supposed to detect those. Okay, so is everybody cool with this? Yeah.
So when it's single positive, I guess. Is it only CD3 and CD4, and it's only CD8 when they detect the pathogen?
Okay, single positive is going to keep one of the two co-receptors that is either going to be CD4 or CD8. Yeah, either one or the other. CD3 is always there. After the double positive stage, CD3 is always there. That's a marker of T cell.
Okay.
Yeah. Okay. Any more questions in here? No, you cool? Okay. All right, so here is summary for the T cell development, right? So first you do the beta and then the alpha, positive selection, negative selection, and the thing goes out. All right. What else is happening here? Things that sometimes are easy to fly over the head because it's just so much information, you drive anybody crazy, right? So what is happening here? First stage of beta chain recombination. What is it that you're recombining? You know, for the cells to stay alive. Have you did the DJ dance? Oh, you can tell by the way I use my walking moves. DJ first, right? DJ for your beta. Okay, DJ goes first, recombines. After successful DJ recombination, you're gonna get your variable region right there. So you pick one of the variable and you put it there. After that, this is where you are not recombining stuff anymore, because you only need recombination for the part of the chain that is going to find antigen. Okay, so that's the part of the beta chain. Let's write the beta chain right here. Okay, the part of the beta chain that recombines is going to be up here. The constant region for the beta chain is constant. That's why it's called the constant region. It does not recombine. Look, you just add a constant region. One of the genes of your constant region. You have several genes of those, but you just put in the constant region. You plug into the stuff that got recombined. Okay? So constant region, you don't have all these rearrangement things. Okay? And this is the same for T cells as well as the B cells. The constant region is not recombining. There's no need for that. Okay? Everybody cool with that? Okay, add the constant region and then you have your beta chain. And the constant region is what is going to be embedded in your membrane. So that's your double layer here, your bilayer cell membrane. Okay? Keep in mind that the part of your receptor that is protruding to the cytoplasm, this part here is very short. Okay, so keep that in mind. I'm going to look at it later. Okay? All right, so here the recombination. Again, this is another picture showing what's happening to your T cell, right? So here you have your beta with the testing pre-T alpha, and here you have CD3 already being expressed, right? And you're going to have your double positive. You have CD, yeah. You can say they all come together, but it's fine. CD3, CD4, CD8, they all get expressed at the same time. Here you have your double positive. Then you're going to have your positive selection. Always positive happens first. Then your negative selection. And then you're going to have your single positive cell that is going to come out and go to the periphery. Okay? All right, so that's the time you stop, you breathe, and you start again. All right, everybody's cool with the T cell? Yeah. You can get it at several stages because there are many. Okay, the question is, where do you get autoimmune diseases? That one, APC1, is happening in the thymus when you are undergoing negative selection. Okay? So that's an important question because I love to ask that question in case of tests. Right? You can thank your friend. Yes?
So what determines the CD4 and CD8?
What determines whether the cell is going to keep one or the other is who is faster at interacting with the MHC that is close by.
Okay.
It's just chance.
Okay.
Yeah.
Okay.
So, yeah, that's pretty much it. And then the other one gets turned off.
Okay.
All right. Well, everybody cool with the T cell? Okay, that was the simple guy. I know. B cells are a lot more interesting. Okay, here we go. Let's go. All right, so B cell. Okay, what's happening here? B cell and surface markers and transcription factors that are going to make this a B cell. Right? So it starts out just like the T cell as a stem cell that is a progenitor. And then at this stage, it is exposed to those same transcription factors. Do you remember them? Interleukin 7 and c-KIT. Yeah, c-KIT. At that stage, because we're going to see the cell is going to proliferate, and then it's going to become a pro-B. After pro-B is going to be pre-B. After pre-B is going to become an immature B cell. After that, it's going to mature. But then the full stage of maturation is not going to happen in the bone marrow. It's going to happen in a secondary lymphoid tissue. Okay? We're going to see how that happens here. All right, so this slide is slightly different than you had before because your very important transcription factor for differentiation of B cell, mainly to keep them stuck as being a B cell for the rest of their lives, that guy is called PAX, PAX5. Okay, so what's happening here? So you have the B cells that are interacting with stromal cells in the bone marrow. Okay, so those stromal cells, you have a lot of them, they're mesenchymal stem cells, right? So these guys here are producing these factors. They are interleukin 7, and there is a C before this kit, so c-KIT, okay? And those are good for lymphoid progenitor cells for both B and T, okay? Then you have transcription factors in the B cell that start to get upregulated. This is EA2 and EBF1, okay? But the thing that really gets the B cell fate really established is the PAX5. That's going to lock this progenitor in a B cell identity. Okay, so that's the PAX5. Now, another transcription factor is important for transition of pre-B to immature B cells, and we're going to see that later. That's a FOXO1 transcription factor. So what's important here? This guy is common. This guy here, he starts getting the B cell into becoming a B cell. This guy locks B cell in the identity of being B. Later on, this guy is going to help this thing to become immature, almost mature. Okay. All right. Let's see what's happening here. So the question here was a big question that was going on for a long, long time and just got solved around 1970. So I'm assuming that's the time when most of your parents were born, which is not too long ago, right? So what's happening here? We know just by looking or examining antibodies, which are proteins, right? Antibodies are proteins. If you get a person's antibodies and you sequence the protein, you're going to see that you have a lot more antibodies that are also B cell receptors, right? You have a lot more antibodies in the blood than you have genes to produce those antibodies. Okay, look at this. The number of antibodies exceed 10 to the 7. That's like a lot of genes, a lot of proteins. What is 10 to the 7? Okay, this is 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 1, 2, 3. This is 1,000, 10 million? Yeah.
When you say there's 10 to 7 antibodies, are those all different types of antibodies?
Yeah. Otherwise, it would be just one in multiple times. 10 to the 7 different antibodies on the cell surface, on the B cell surface. Do you know how many genes you have that express protein that comes with your genome? Let's take a guess. Genetics, anybody? Okay, who took genetics? I want to see arms up. Who took genetics? And they didn't tell you? Who you took genetics with? Okay. Take a guess. Guess. Number of genes. Any number. Brave souls. Cricket. Cricket. I don't like to pick on people. Too many. But that was a good guess. Because it would be about half. It would be about half, but it's not. It was too much. What? Yes! Somebody pay attention to genetics. Was it a guess or you just look it up?
I just read it.
I know. you guys think that we never see what's happening. But that's fine. That's fine. 25,000. About 25,000 genes in your whole DNA. All of the chromosomes that you have, you express about 25,000 protein genes. How many gene products in general for your somatic cells, they are not B or T cells, how many gene products do you have expressed in your cells? Do you know that one? You can look it up, it's okay. How many genes are expressed in a somatic cell? More than 25,000? Guess?
50,000?
A little bit more.
100,000?
100,000. About 100,000. Look at this. let's put a different color here. 100,000 genes are expressed in your cell. Do you see a problem here?
Yeah.
This is your first problem right there. Your DNA is only expressing about 25,000, 30,000 at the most. Your cells express 100,000. Where are the other genes coming from? Those cells are not recombining. Those are irregular somatic cells. That problem is still being solved. Those 75,000 missing genes are coming from your microbiome. Cool, huh? What you eat and what you do with your life and lifestyle is going to dictate the genes that are expressed in your body besides your own DNA. Okay, that is called epigenetics. We're not talking about epigenetics here. That's a different problem, right? There is this book called Missing Genes that if you're interested in this case, you can read it. I didn't read it yet. Okay. Now, we're talking about this other problem here. 10 million different antibodies. It's even worse than the missing gene problems, right? So now scientists are starting to question, like, how can you produce 10 million different B cell receptors if you only have about 25,000 genes in total? And let's say if you use all of your genes to produce B cell receptors, you have no genes left for anything else in your body. So that's even, that's a worse problem, right? So the question is, how is it that those cells are doing that? Okay. So after figuring out what the problem is, then they had to figure out how to solve the problem. So they quickly realized that something is happening with the heavy chain and the light chain of those immunoglobulins, that they could have extraordinarily variable sequences, right? So they were getting recombined. So they really thought about recombination of gene pieces, okay? And another idea that came up was that the different parts of the immunoglobulin would have to come from different chromosomes, okay? So here you have your germline DNA. Okay? So you have a chromosome here, and there, and other chromosomes here. So heavy chain would come from this chromosome, light chain would come from a different chromosome, okay? And they also realized that the B cell DNA is different from a germline DNA or from a stem cell, because your B cell DNA is going to be shorter than the germline DNA, okay? So what you have here, you have heavy chain and light chain coming from different chromosomes, and if you look at the total DNA, your B cell is going to be shorter, meaning that it lost genes when you compare it to the germline DNA or to any of the other somatic cells that you have. So your B cells have lost genes. So this is the part here, okay? Besides that, around the 70s, they finally figure out what is happening to add variety to your antibodies. It was this thing called somatic hypermutation, okay? Because this is not just recombination. Recombination, you get the gene, you put it there, you get a gene, you put it there, you recombine them, independent terms, so and so. But it's still that did not explain the variety of antibodies. When they figure out about this somatic hypermutation, then they could figure out what's going on with those antibodies, okay, with these B cells. And somatic hypermutation is something that only happens with the B cells. It's not going to happen to the T cell. Okay, only B cell is going to do that. Okay, so let's look here what is happening to your B cell thing. So it starts out with the heavy chain, and we have seen that before with your T cell, right? So this is your B cell. First thing that's going to happen with your heavy chain, so here you don't call beta, you call it heavy, HC, okay, heavy chain. First thing that's going to happen, same story as with your T cell, you get your DJ recombination. Then you're going to get your VDJ, then the recombination is going to stop, and you're going to add your constant region. Again, the constant here is not going to recombine. the first constant region that is going to be joined to all the stuff that got recombined, the first constant region is going to be your C mu. That's always the case. Okay? So C mu is going to be the first constant, and the constant region is going to determine the type of antibody that you have. Okay, so the first one is always going to be C mu. The first antibody you produce is always going to be membrane-bound M antibody. Okay? Okay, so this is the story with the heavy chain. And then for the light chain, same thing that happens, but you don't have the D. It's lighter because it doesn't have D. Okay, it's smaller. So you get your VJ, your combination, and then your combination stops, and you get a constant region right there. The constant region of your light chain is not going to dictate anything about the type of antibody. It's just there. Okay? So the one that is really going to determine is the constant region of your heavy chain. Okay? And that is important. Okay? Because we're going to see how that's going to change later. All right. So here is a little exercise that is going to tell you what are the possible combinations that you can get just by recombination alone. So combinatorial antibody diversity. Okay? So if you look at your heavy chain number of segments that you have for each of those genes, so remember DJ happens first, right? And then you get VDJ. Okay? So if you're going to combine D and J, you have 23 picks for your D. Okay, so you have a paper bag, 23 different genes in there. You're going to pick one. After you pick a D, then you're going to get a J. You have 6 possible combinations, 6 possible genes that you can pick for J. You get in another bag, you pick up another ball, a J ball. Okay, what is the total number of combinations that you can get only from D and J?
138.
So we multiply 23 by 6. Everybody got that? Yeah? Is that simple? Who didn't get that and we need more explanation? Okay. All right. So simple. You get those two terms, you multiply them, and you get 138, something like that. Now, after this, then you're going to get your variable region. So now you're going to get here and you say this number times 6, because another independent term. So only by recombination you can get all this number of different heavy chains. Okay? And we didn't even start talking about those T enzyme stuff. Okay, you're going to give it even more recombination possible, even more variety possible. Okay, so that's the story with your heavy chain. Now let's see what happens here with your light chain. Okay, keep in mind for the heavy chain, you have it in a chromosome that you inherited from dad, and then you have another chromosome that you inherit from mom. So if the combination from one of those was totally messed up, you have another chance to get it right with your other chromosome. Okay. When you look at the light chain, you have more chances of getting it right. Okay. So for the light chain, you're going to have a kappa gene that is going to be in one of 2 chromosomes, and you're going to have a lambda gene that is going to be in also one of 2 chromosomes. Okay, so you got 4 chances to pick the right light chain. Okay, and again here you can just go through the math and see what are the possible different combinations that you can get from that. Okay, is that simple? So if I ask you something giving you a certain number of genes, and can you figure out what is the total combination you can possibly have? That is always a good test question, right? Okay, so here we go for some more. Okay, so this is actually the same thing, right? So what is saying here, just for your studying, okay? So you have here your independent variables, right? I just put in words what I just talked about right now. Okay. Okay, so here we go. All right. Oh, first question, finally. If you want to alter the antigen binding site of an immunoglobulin, what are you going to mutate? So now you're going to think antigen binding. You have to think about your antibody shape. What is going to recognize antigen? In 5 seconds. All right, does anybody need more time? Yeah, I'm stopping it. What is your answer? Is D. You're going to have the variable region, right? Because the constant doesn't change. You're going to have the variable region mutated. All right. Okay, so let's see here. You know, to make your antibody, you're going to have 2 identical heavy chains. So here is those are your 2 identical heavy chains. And you're going to have 2 identical light chains that are going to interact with those heavy chains, right? The light chains can be of one of 2 flavors. Either they're going to be both kappa or they're going to be both lambda. So let's give lambda a different color here, okay? Either both kappa or both lambda, not one of each, okay? And we're going to see why. Now, what is happening to those genes over here? The light chains are encoded in different chromosomes. How many chromosomes do you have in total? 46, right? 23 pairs, right? So here you have 2 chromosomes that are going to come from, one from mom, one from dad. And here you have another chromosome. So this guy here is a big chromosome, right? Because your lower number are the bigger ones. This one here, look at this, chromosome 22, all the way close to your sex chromosomes. That's the 23rd pair, right? And then you have your heavy chain still in another chromosome, okay? So this is important for that recombination type of event that goes on, okay? So let's see what is happening here. With your common lymphoid progenitor cell, now you're going to have your full development of your B cell. That's going to go through 6 different stages. Now look at this. First thing is going to be an early pro-B cell. Then you're going to have a late pro-B cell. Then you're going to have a large pre-B, a small pre-B, and finally an immature B cell. Okay, it's going to mature in the lymphoid, not thymus, in the lymphoid or secondary lymphoid organs. Okay, so 6 stages. Every time you are going through a different stage, the genome of this cell is losing a piece of DNA. Okay, so the DNA that is going to be here is going to be shorter than the DNA that was over there. It loses a piece of DNA at every stage. Okay. All right. So let's see here. Heavy chain, light chain. So this we have seen before. All right. So the heavy chain, this is important. The heavy chain is going to determine what kind of antibody that you're going to have. Okay. So you have how many types of different antibodies I will see before? 5. Okay, so do you remember what they are? IgA, IgM, E, and IgD. You have 5 different types of antibodies. Your heavy chain is going to determine which antibody you're going to have. Okay? All right, so let's see here. What can those antibodies bind? Remember, they recognize 3D. They're going to recognize proteins and carbs or lipids, proteins or carbs or lipids. Okay, they can organize all this kind of stuff. Okay, they have a diversified way of getting to recognize things and getting activated. Okay, so now let's see here. What happens? This story of allelic exclusion is the same thing as we have seen before with the T cell. Okay, so this is going to happen for both of the heavy chain and the light chain. Okay, remember, here is a little bit different, the story of the heavy chain of your B cell. Okay, because what's going to happen here with the B cell, you're going to start recombining both of the genes from both chromosomes of your parents. Okay, so you have your heavy chain, you have the heavy chain here in these two chromosomes. Both of them is going to start recombining. Once one of them gets a good recombination for the heavy chain, then it's going to stop the other one from continuing recombination. Okay, so start together and then they're going to stop the other one. So it's going to shut down the recombination of the other guy. So that is what is called allelic exclusion. Okay. All right. So remember then, once you make the heavy chain, then you are going to start testing it, right? So you're going to need your surrogate light chain to test it, right? And we're going to see this with pictures, because if you just read this, it's just very confusing. All right. So here it is. Okay. So what's going on here? Stages of your B cell development. So here, remember, you started at the same stage as your T cell. Everything here is happening in the bone marrow, so it's not going to leave the bone marrow. Okay, so your stem cell, first thing that's going to happen here is not showing transcription factor here, but you do have IL-7 and you have c-Kit, right? c-Kit is being expressed there. Okay, and then you're going to have proliferation of your B cell. Once you get to a pro-B stage, you are going to stop proliferation, and then it's the same story. You're going to start upregulating here your RAG genes for a combination and your TDT genes for your heavy chain, right? At that stage, when you have your heavy chain expressed, you get to be pre-B. Okay, pre-B, you have heavy chain, but you don't have the light chain. So you're going to have the heavy chain and you're going to have your surrogate light chain, right? You're going to test your heavy chain. Once your heavy chain is tested and is good, you're not going to go straight to immature. First thing you're going to do, you're going to shut down those recombination genes, and you're going to proliferate that heavy chain. Same thing that happened with your beta chain. Proliferate, because now all the daughter cells will have a chance to get different light chains. And then what's going to happen? You stop the proliferation, you're going to upregulate the recombination genes for your light chain, you're going to recombine the light chain, you're going to turn off this gene, and now you're going to have your mature B cell that has your heavy chain and light chain on the surface. Oh, there is more to this, okay? This is your immature B cell that already has that heavy chain and light chain on the surface. Okay, something is missing here, so pay attention. Okay, because none of the immunology books that I have looked so far is totally perfect. Okay, so what's happening here? Your immature B cell makes that first type of antibody that is your IgM bound on the surface. Okay, once that IgM works, now the cell is going to get a signal to become mature. Now the cell is going to make another antibody on the surface, right there, okay? And this guy here is going to be IgD. So you're going to have IgM and IgD. Now that IgD, the formation of the IgD, is going to tell your B cell that is mature and it can leave the bone marrow and it can go to the periphery. Okay, so development in the bone marrow is done once you get IgD on the surface. Okay, so let's see what happens here. Okay, here are the stages that what you're going to see on the surface of the cells. Okay, Okay, so here's the story with your IgM. Okay, IgM, what is going to happen here? When you have your heavy chain recombined, okay, VDJ, okay. Okay, one interesting thing about the heavy chain is that for the addition of your variable region, you get a VDJ recombinase that is just adding the variable region. Okay, once you have your heavy chain done, then the constant region that is not showing there. Okay, let's put a constant region here. Your constant region here, the first one is going to be mu. Okay, so this is why I have a cow over there. The first immunoglobulin that is made is the mu one. Okay, is the M, mu. So hopefully you're not going to forget that. Okay. Okay, so now what is going to happen here? Okay. Remember when you're testing your heavy chain, the same kind of testing that you have done with the beta chain for your T cells, you're going to have a surrogate chain, right? And this surrogate chain here is going to be your, is just called surrogate chain, but some important things about the surrogate chain. The V3B part of the chain is the part that would bind to antigen. And then the lambda 5 is the part that will be a constant region of your light chain. Okay, so your surrogate chain is made up of 2 proteins. Okay, so you have V3B and lambda 5. Remember, that guy does not recombine. It's just testing your heavy chain. Okay, what else is important here? Okay, I already mentioned that. Okay, all right. Okay, I mentioned the rearrangement and allelic exclusion. Okay, I already mentioned all that. So let's see here. All right, so your RAG genes, DTT genes that are being recombined. Same story as you have seen before, okay? So you have here proliferation, then you have rearrangement, including DTT, heavy chain, then you have proliferation, then you have rearrangement of your light chain, then you're gonna get your mature B cell. Okay. And that, okay. Oh, this guy is repeated, okay. Ignore this guy. Oh, gee, I get them both. Okay, messed up with the arrangement. Okay. Right, so, okay, you tested your heavy chain. Okay, why do you need to test the heavy chain? Okay, let's see here. This is important to have mentioned before. How do you test the heavy chain in the first place? Okay, so your heavy chain is on the surface of the B cell, and when it interacts with antigen, the whole idea of this guy being on the surface is that it's supposed to send signals to the nucleus of the cell to upregulate certain genes, because the cell has to become a plasma cell, right, after it sees antigen. What happens here is you look at your antibody here, and the tail of the antibody that is going through your plasma membrane, that tail is kind of short. So you don't have, the antibody doesn't have what it takes to send a signal down to the nucleus. So besides your B cell receptor, you're also going to have expression of 2 very, very important proteins. That is going to be immunoglobulin beta and immunoglobulin alpha. So you're going to have alpha and beta that are going to be expressed on the surface very close to your receptor, okay, to the B cell receptor. So this alpha and beta, once you have activation due to antigen binding, that this is what your surrogate light chain is doing here, okay? Once you have activation, this is supposed to send a signal that is going to be transmitted from your B cell receptor to this alpha beta, that these guys are going to allow some phosphorylation to happen, is going to send signal transduction to the inside of the cell, okay? So the testing of your heavy chain is whether this thing can interact and activate survival signals that are going to go through the alpha beta, okay, Ig alpha and Ig beta, that's going to go inside the cell. And yes, I know this is confusing because you call beta alpha the receptor for your T cell, right? And this is why I'm allowing you to take the test in pairs. okay, because this thing will drive anyone crazy. Right? And also, all this stuff is important because if you don't have this Ig alpha and Ig beta right there, you don't pass the signal. So if you don't have functional heavy chain, you don't pass the signal. Okay, so everything is important here for you to send a signal for the cell to survive. Okay. So this is on the surface. All right, so once your heavy chain is tested, then you're going to become small pre-B cell stage. That's when you're going to make your light chain. All right, so here, your small pre-B cell, you make your light chain. And what's happening here with the light chain? So here, you don't have both genes that are competing with each other like you had in the heavy chain, okay? Heavy chain genes, both chromosomes started, and they were like running a race to see which one is going to recombine successfully, right? But that's not happening here. So here, you're going to have first the kappa genes that are going to recombine, and only in one of the parent chromosome. Okay, so these guys are more polite. Okay, so you have here both kappa genes, like mom and dad chromosomes over there. It's like, okay, heavy chain is done. We shall make a light chain. Honey, do you want to go first? And they want to say, oh, okay, I'll go first. I'll recombine. I'll recombine. Oh, look, I made a pretty light chain. Guess what? You are allylically excluded. So the guy's not going to work. So one of them worked, everybody else gets allylically excluded. Okay? Now let's say that your first chromosome in your kappa gene didn't work. Now the other one, which is a kappa gene from, okay, so let's call this one was your kappa gene from your mom chromosome. This one here is a kappa gene from the dad chromosome. If the mom chromosome gene did not work, now you're going to try the kappa gene from dad. Notice you did not go to lambda here. Kappas go first, okay? If that one works, then you don't worry about anything. If that one doesn't work, now you're going to start with one of the two lambda genes from the other chromosome. So you're going to have 4 chances of getting this light chain right, okay? Why? Well, because it was a lot of time and effort that the cell already put into making the heavy chain. And if it just dies at the first trial here, then it's a waste of energy. And cells don't like to waste energy. Okay? So 4 different chances of getting this light chain right. Okay. Primary, okay. Nascent peptide, okay. Kappa light chain. All right, so this is just a story with your kappa rearrangement. Okay. Oh, another question here. Not an easy one here. The kappa and lambda genes, what are they referring to? Easy one. You don't need more than 30 seconds for that. So, 10 more seconds. Do I need more time? No? Okay, what's your answer? Yes. What's your answer? B. All right. So that's the light chain. Now, when you have your heavy chain and your light chain working, okay, so now you gotta test your whole antibody to make sure that it does not bind self, okay? So this is the time that your cell will go through negative selection, okay? That's similar to what happens to your T cell, except that here your B cell doesn't have the positive selection. Okay, so it only has a negative selection. And another thing that is different, the stromal cells in your bone marrow, they do not express the ARI gene. Okay, so the self-antigen buffet that these B cells are getting is different than the buffet that your T cells get in the thymus. Okay, you have less diversity of self-antigens that is happening in the bone marrow when you compare it to the thymus diversity. Okay, but it's still good enough that you do not get a lot of self-reactive B cells out there. Okay, all right, so what is happening here? In the bone marrow, you have your immature B cell that has your heavy and light chains right there. And here is your alpha and beta, Ig alpha, Ig beta. Okay. If it gets a go ahead and did not get killed by negative selection, right? So what's going to happen is that you're going to have alternative splicing of your RNA that is making your IgM. Okay, I know there's a lot of stuff in there, a lot of information right there, okay? When you get your alternative splicing, you are going to get IgD on the surface of your cell, and that is going to tell your cell that it is mature, okay? Mature but naive. Once you get IgD expressed, then the cell leaves the bone marrow and goes to the periphery. Now, how is it that this is happening, okay? Look. When you have your constant regions, the first one, remember, of your constant region that goes is the mu, right? Now mu, it doesn't go alone. It goes mu and delta. They go together, okay? When you make your RNA, you're going to have a site that is going to tell the RNA that it can do alternative splicing to either express the mu gene or the delta gene. So with this RNA alternative splicing, it can splice out this exon of mu. Then it's going to express the delta gene. And this is why you end up with both IgM or IgD on the surface, because the constant region is what's going to tell you what kind of antibody you have. Okay? There is more to this story, but is everybody okay with that? Yeah, okay. So, yes, no, breathe. Okay, now the B2 cells that you have in circulation in your blood, in the body, okay, they have a lot more IgM than they have IgD, okay, but they have some IgD. Okay, so let's see here, more explanation of what is happening to that RNA, okay. When you look at the constant regions, okay, you know the sequence of these constant regions, it goes like this, okay, mu delta, gamma gamma alpha, gamma gamma, epsilon alpha, okay, I don't care. But here you have mu and delta close to each other. And do you see something different? Well, maybe you don't, but I'm telling you. You see this little dot right there? Before the mu, that's a switch signal, okay? And then you look here and you have on gamma, gamma alpha, blah, 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 blah. All the other ones have a switch signal, but the delta does not have it, okay? When you make the RNA, okay, this is the DNA. When you make the RNA that has recombination here, you're gonna go to the first constant region, you're gonna make RNA that has both of these pieces in it, okay? So that primary transcript is going to have mu and delta with it. And I have a better picture for it. Here it is, okay? That primary transcript is going to have mu and delta, okay, that come together with the RNA. So this is RNA, okay? When you do RNA processing, I know everybody took cell bio, right? RNA processing, remember that, the pain in the neck? Splicing stuff, okay? So in this case, you can splice out an exon of the mu. If you splice out mu, you end up with the constant region that is delta. So this is going to be your delta RNA, okay? So that's going to allow expression for IgD or IgM on the same cell. Okay, remember both of those have the same binding for antigen. They have the same antigen binding site. Yeah. The IgM only has the mu because when you are expressing from RNA to protein, you're going to express only the first one that is on that sequence. Because look at this. That's a good question, actually. Okay? The question was, does the IgM has mu and delta or just mu? You see this little dot right there? This is a signal for a poly-A site. Okay, so the primary transcript on the mRNA, it has both of them, but it's carrying with it this poly-A site right after the mu. So if the mu is not spliced out, then you're going to make the poly-A tail, and that guy is never going to be expressed. Okay, that was a very, very good question that sometimes I forget about it. But I'm glad you reminded me. Okay, so look, poly-A tail is making sure that this RNA is going to be stopped right there once it gets translated, right? Once it gets translated. Okay, now if you splice it out, then your delta here, look, is going to have your poly-A attached to it, right? And then you're going to have your IgD antibody on the surface. Okay. Is everybody cool with that, with this mechanism? That's a hot topic mechanism because it's the one that is different. Okay? All right. Okay, so then what's going to happen? Once your B cell goes to the periphery, it is going to go to the secondary lymphoid organs, and it's going to sit there until it gets activated. There are several ways, as we're going to see, that you can activate a B cell, but the really good one is when you use a T cell that is going to interact with a receptor on the B cell. Okay, so here you're going to have your T cell receptor that is going to bring an antigen and interact here with your receptor in the B cell. Okay, once T activates B cell, you're going to have this protein on the T cell that can interact with a receptor on the surface of your B cell. Okay, so this guy on the T cell is called CD40 ligand. The guy on the B cell is called CD40 receptor. Okay, and yes, those are important because if they interact, you're going to send signal to the B cell to behave differently. So by T cell activation, your B cell is going to start proliferating a lot and is going to further differentiate into a plasma cell. Okay? And depending on what your T cell has seen, whether it is a virus or a bacterium or a fungus or who knows what, depending on the signal that the T cell got at first, is going to secrete different cytokines to activate your B cell, and that is going to tell the B cell what kind of antibody is going to produce. So depending on what signal T cell got is going to teach the B cell on how to further differentiate. Okay, so there's more story to the B cell. All right, so what is the B cell going to do? It can secrete antibodies that are of IgM. So that's the first antibody that gets secreted. Okay, so that's your IgM. Remember, when IgM is secreted, it's not a monomer anymore. It becomes a pentamer. Okay. Now, instead of IgM, it can secrete antibodies that are IgG or IgE. Those are monomers. Okay. Or it can even secrete antibodies that are IgA. Now, this type of dimer IgA is important for mucosa, or you can also have a monomer IgA that is important for your circulation. Okay, so here you can see where these antibodies are going to come from, but that is going to be after your T cell CD40 ligand interacts with the B cell. Okay, all right, so let's see what else here that is important. All right, so we went through this. Oof, all right. And yeah, we only have like 6 minutes until this time is over. We are going to look at this next class, but make sure that you watch those extra videos too, because they are very informative, okay? Before you start running out of the class, do you have questions? Everybody's cool? All right.